Who will tell reason for observation? Hurry fast. Yes. Arushi Singh and Saksham Barwal. Ma'am, please unmute them one by one.
Sir, the reason for this observation is that the germinating seeds, as they respire, they acquire the oxygen present, and as the experiment is airtight, so to fill the space, so to fill the vacuum, the water rises up from the beaker.
Okay. All right. Yes, Saksham. Unmute yourself.
Yes, sir. The germinating seeds will produce carbon dioxide. The soda lime will absorb carbon dioxide, and therefore there will be a gap. I mean, there will be no air. Therefore, a vacuum, the kind of vacuum, will be created, and it will
All right.
rise, taking the
Okay,
level.
good. Now listen now. Listen everyone. We have to give the reason for observation. So look at here. Here is a germinating seed. It means they will germinate. Germination will take place only when there is an oxygen here. So whatever the oxygen is present in the conical flask, that will be consumed by germinating seed for respiration. First point, germinating seed will consume oxygen present in the conical flask. When they germinate, respiration takes place. So what will happen? Carbon dioxide will release. So carbon dioxide will be filled here. But KOH is here, which is an absorbent of carbon dioxide. It will absorb carbon dioxide. Thus, vacuum is created there. So the water from the beaker rises in the delivery tube. So how you will write? Write it there. Seeds respire aerobically. Germinating seeds respire aerobically. Consume oxygen of the flask. They will consume oxygen of the flask. This flask have oxygen. So reason for observation, germinating seeds respire aerobically, consume oxygen of the flask, and produce carbon dioxide. And produce carbon dioxide, and this carbon dioxide is absorbed by the KOH solution. And this carbon dioxide is absorbed by the KOH solution, creating a vacuum in the flask. How the vacuum is created, now you know that. Creating a vacuum in the flask, as a result of which the water of the beaker rises in the delivery tube. The water of the beaker rises in the delivery tube. Again, I'm repeating, germinating seeds respire aerobically and consume oxygen of the flask and produce carbon dioxide, which is absorbed by the KOH solution, creating a vacuum in the flask, which results in the rise of water in the delivery tube from the beaker, which result of which the water of the beaker rises in the delivery tube. Got it clear, everyone? Next, conclusion. Conclusion. What will you conclude? So whatever is the aim, that will become the conclusion. Aim was what? To demonstrate that aerobic respiration in germinating seed. Now you will conclude this experiment prove that germinating seeds, aerobic respiration in germinating seeds. This experiment demonstrated that germinating seed respire aerobically, consuming oxygen, producing carbon dioxide. Got it clear? Now, how the board asks you the question. How the board will ask you this question. Everything you have understood, aim, procedure, observation, reason for observation, conclusion. Now, how board will ask you? Board will give you this diagram. Note my word. Diagram given in an experimental setup to demonstrate a physiological process in plant. Diagram given in an experimental setup to demonstrate a physiological process in germinating seed. Now here the germinating seed, it will not be a plant. What will give you this diagram? The diagram given in an experimental setup to demonstrate a certain biological process in germinating seed. Finished. First question, what is the aim of the experiment? Second, name the process demonstrated. First question, aim of the experiment. After giving this diagram, nothing they will give, only this diagram. Second, first aim of the experiment. Second, name the physiological process. Or name the process demonstrated. What will be the name of the process? Yes, who will tell? Fast. How much time you will take? All these are the board question only. What you will like? Name the process demonstrated in this experiment. Which process we are talking about here? Yes, unmute yourself. Anjali, I think.
So respiration.
Respiration. Either you like respiration or aerobic respiration because you have proved that oxygen they have used. So you will like respiration or aerobic respiration. Both are acceptable, but not anaerobic. Third question. Define the process. So you will define the process. Fourth question. Give an overall equation of this process. So you can give an overall equation for this process. I have already mentioned. Fifth question. What will be your observation? I have dictated the observation. Next question. Give reason for your observation. Give reason for your observation. Next question. Why the seeds are soaked in disinfectant? Why the seeds are soaked in disinfectant? To kill the bacteria. Because we have studied today only bacteria and fungi. They go for anaerobic respiration. That's why seeds are soaked in disinfectant to kill the bacteria. Next question. What is the purpose of using KOH solution? To absorb carbon dioxide. KOH is an absorbent of carbon dioxide. To absorb carbon dioxide. Next, what will be your conclusion? That also we have done. And the last question, suggest a suitable control for this experiment. Suggest a suitable control of this experiment. What is the control of this experiment? The same experimental setup, everything will remain the same. Instead of germinating seed, take boiling seed or dead seed. The same experimental setup with boiling seeds or the dead seeds. That will become the control of the experiment. Now heat is given out. To demonstrate that heat is given out during respiration in germinating seed. Now, method. In this, what you have to take a thermoflask bottle. Take a thermoflask bottle containing germinating seed or live seed soaked in disinfectant. All right. And you put some germinating seed here. Soaked in disinfectant. I have told you the reason also why you will soak this germinating seed in disinfectants. Put it. Now this is germinating seed. This is thermoflask. Now introduce a thermometer inside the flask through the hole of the fork, through the hole of the cork fitted into the mouth. Now cork here we have put a cork, and through one of the cork we are putting the thermometer. We are introducing here the thermometer. With the help of hole of the cork, introduce the thermometer. Fit it into the mouth of the thermoflask. Now the bulb of thermometer, this is the bulb of the thermometer. The bulb, this is an important point. The bulb of the thermometer must be in contact with the germinating seeds. Must be in contact with the germinating seeds. All right. This is the bulb. So this has to be contact. This bulb must be in contact with the germinating seeds. And earlier, this is the bulb of the thermometer. So superficially, not above or just above, you will not put this thermometer, the bulb of the thermometer. It should be deeply placed in the germinating seed. That's why I have made it here, the bulb of the thermometer. And this is what we have, thermometer. Now, what will you observe? This is the method. take a thermoflask and put some germinating seed soaked in disinfect. The first thing. Alright. Then introduce a thermometer with the help of hole from the cork into the thermoflask. And the bulb of the thermometer, this bulb of the thermometer must be in contact with the germinating seed. These are the points. Now, what will you observe? After aim observation method, observation. Temperature in the thermometer rises. Observation, temperature in the thermometer rises. Temperature increases. Now, how you will give the reason for observation? Next, reason for observation. Germinating seed respire and produce heat energy. And this heat energy causes the temperature to rise in the thermometer. This is the reason for observation. Again, I am repeating. Reason for observation, germinating seeds respire. Respiration will take place in germinating seeds. Here we are not talking about aerobic or anaerobic. Here only respiration is there. All right. Here the respiration is taking place in germinating seed. Germinating seed respires and produce heat energy, and this heat energy causes the temperature to rise in the thermometer. Now, last question. Why is the bulb of the thermometer must be in contact with the germinating seed? What is the reason behind that? Yes, who will tell? Why this bulb of the thermometer must be in contact with the germinating seeds? Yes, anyone? All right, write it there. Because in respiration, write it fast, hurry up. Because in respiration, heat produce is given out to the all these are the board's question only. Because in respiration, heat produces is given out to the surface of the seed. And make sure you all are writing. Because in respiration, heat produced is given out to the surface of the seed. Done. This is the aim of the experiment, observation, reason for observation. All right. And the conclusion. Now, next experiment number 3. Same, they will ask you the question. The diagram given in experimental setup to demonstrate certain physiological process in germinating seed. Name the process. Now here, what process will you write? Respiration. All right. Name the process respiration, because heat is given out in both the process, whether it is aerobic or anaerobic.